Every second, laboratories across the globe generate terabytes of genetic information, creating a constant, invisible flow of biological data. To manage this volume, we have to look past the global network and zoom in on the physical source, the cell. We categorize this information by its cellular origin. Sequence data provides the raw genetic code. Structure data provides 3D atomic coordinates. Expression data measures transcription levels. Interaction data maps signaling networks. And phenotype data records observable traits. Tracking these 5 distinct categories across millions of organisms creates an immense storage and organization challenge that requires a specialized digital architecture. Without this architecture, raw genetic code is trapped in isolated laboratory silos. This unmanaged data leaves researchers with a tangled web of disconnected information. Biological databases solve this.
They centralize scientific knowledge and enforce universal formats, ensuring every sequence is machine-readable and properly annotated. This infrastructure is what transforms raw experimental output into a searchable, actionable body of scientific knowledge. Biological information originates from 3 primary pillars: physical wet labs, public open access vaults, and computational dry labs. The life cycle begins when a researcher extracts raw DNA from a physical organism in the lab. To move that sequence into the global system, the scientist must upload it using specific submission architectures, such as Bankit or Sequin. Bankit is a web-based tool for simple, single sequences, while Sequin is a standalone application designed for complex whole genome submissions. This submission phase effectively converts a perishable physical sample into a permanent digital asset ready for global review. Once submitted, raw experimental sequences enter a primary database archive. Data then passes through a curation filter, where scientists verify and annotate sequences into an organized secondary database. Finally, composite databases link these curated sources, allowing complex searches across the ecosystem. This table highlights the difference between the tiers. Primary databases often contain high redundancy from original submitters, while secondary databases are consolidated and peer-reviewed by expert curators. This filtration pipeline ensures that raw, overlapping genetic submissions are eventually refined into an authoritative reference. For this vast data to be useful, scientists around the world must be able to query the global system and extract specific records for analysis. Retrieval follows 2 primary topologies. NTRES functions as a centralized hub linking records within the NCBI ecosystem, whereas the Sequence Retrieval System, or SRS, links disparate databases across the globe. Even after retrieval, scientists face an interoperability problem because different software tools use different digital languages. Conversion tools act as translators, extracting the core biological data from one format and restructuring it into another to ensure a seamless flow into analytical software. For example, the GenBank format contains rich, human-readable metadata, while the FASTA format provides a streamlined computational string designed for rapid alignment. Standardized formats like FASTA allow global laboratories to analyze sequences using a common digital language. This blueprint represents the complete life cycle, tracing a sequence from its generation in the lab to its final processing in computational software. These databases function as active, interconnected ecosystems. This architecture exists as a live repository that drives global scientific discovery. This data flow powers modern advancements, such as AI-driven protein prediction models and the rapid mapping of complex diseases. Progress in personalized medicine and agriculture depends on the ability to maintain and expand this shared digital infrastructure. The biological database is a collaborative achievement. It ensures that every new scientific breakthrough is built upon a foundation of shared global data.